Hello everyone, welcome back to my YouTube channel. Today topic is about the hypersensitivity, particularly hypersensitivity 1. So first, what is hypersensitivity? Hypersensitivity, also known as allergy. Okay, it happens when the immune system overreact to something that is usually harmless. They are usually referred to as an overreaction of the immune system, and this reaction may be damaging, uncomfortable, or occasionally fatal. Okay. So refers to undesirable reaction the normal immune system produces, including allergies and autoimmunity. Hypersensitive reaction require a pre-sensitized immune state of the cells. Okay. So what is the pre-sensitized immune state? The term pre-sensitized state or immune sensitization refers to the condition in which an individual immune system has been previously exposed and has developed a specific immune response against a particular antigen or allergen. Okay, so in the context of hypersensitive reaction, especially type 1 hypersensitivity, this pre-sensitized state involves the presence of specific immunoglobulin E, IgE antibody. Okay, so now the allergy, it is a type 1 hypersensitive reaction. So more than half of the US population, that is the 54.3%, suffer from the type 1 hypersensitivity reaction, which is encompass the most common allergic reaction, including the hay fever, asthma, atopic dermatitis, also known as eczema, and allergic conjunctivitis. Right? So here it is the word that is atopy. Atopy refers to the genetic tendency to develop the allergic disease such as allergic rhinitis, hay fever, asthma, and atopic dermatitis. And atopic, okay, both are different, atopy and atopic. The atopic refers to the predisposition to develop allergic condition. For example, deposition of IgE antibody within the plasma. Okay, so first disease, that is the allergic rhinitis, also known as the hay fever. So in this disease, inflammation of the nasal passages due to the exposure of allergens like pollen, dust, and pet dander. Now second one, that is the asthma. As we know, asthma, chronic respiratory condition characterized by the inflammation and narrowing the airways and leading to difficulty in the breathing. Third, that is the atopic dermatitis, eczema. So a chronic inflammatory skin condition characterized by the itching, redness, and the development of rash-like patches. Fourth, that is the allergic conjunctivitis. Okay. We very well know the conjunctivitis of eyes. Okay. Inflammation of the conjunctiva, the thin tissue covering the eye due to the allergen exposure causing redness and itching of the eye. Okay, so IgE antibodies are responsible for the type 1 hypersensitivity. Okay, so this is the picture in which this is the antibody and that is the antigen. Okay, type 1 hypersensitive reaction, also known as the allergies, are initiated by the interaction between IgE antibody and multivalent antigen. Multivalent antigen is an antigen that has multiple binding site. It is, or we can say, the epitopes capable of interacting with the antibodies or immune cells. Okay, so here it is, the many allergens can elicit the type 1 response. Healthy individual generate IgE antibody only responds to parasitic infection. However, some people referred to as atopic are predisposed to generate IgE antibody against common environment antigens such as those listed in this table. Okay, for example, plant pollens, ryegrass, ragweeds, timothy grass, beech trees, food like nuts, seafoods, eggs, peas, beans, and milk. They have the allergy. Okay, and they produce the type 1 type of hypersensitivity. Drugs, penicillin, sulfonamides, local anesthetics, salicylates, insect products, okay, bee venom, wasp venom, ant venom, cockroach galaxy, dust mites, mole spore, animal hair and dander, latex, foreign serum, and vaccines. Like all are the allergens, okay, which produce the allergy. Okay, now the IgE antibody act by cross-linking Fc epsilon receptor on the surface of innate immune cells. So the in normal individual, particularly the level of immunoglobulin E in the serum, that is the liquid components of the blood, is relatively low compared to other types of immunoglobulins. Normal level of IgE in blood can vary, but they are generally in the range of 0 to 100 international unit, okay, IU per ml in adults. Okay, so IgE antibody are not destructive, alone not destructive. Instead, they cause hypersensitivity by binding to particular receptor, that is the Fc receptor, okay, which is present in mast cell and basophils and also in other cells. These are expressed by variety of innate immune cells including mast cells, basophils and eosinophils. Now the binding of IgE antibody to Fc epsilon RS, okay, receptors activities these granulocytes which are present in mast cell or basophils, release the content of intracellular granules into the blood. Okay, and this process called the degranulation. The content of the granules vary from the cell to cell, but typically include the histamine, heparin, and proteases. Now, together with other mediators, leukotrienes, that is secondary mediator, prostaglandins, chemokine, and cytokine. This mediator acts on the surrounding tissue and other immune cell like neutrophils, eosinophils, causing allergy symptoms. Once the IgE antibody binds to the Fc epsilon receptor, which is present in the mast cell, basophil, eosinophil cell surface, okay, and leads to the degranulation. And these granules contain the primary mediator and secondary mediators. So primary mediators, that is example histamine, heparin, okay, normal, and secondary mediators is leukotrienes, prostaglandins, chemokine, and cytokine. Now here it is the structure, how the produce the mast cell, or how mast cell degranulations occurs. Okay, so general mechanism underline an immediate type 1 hypersensitive reaction. So first here it is B cells. Allergen, okay, exposure to an allergen activated Th2 cells that stimulate B cells to form IgE secreting plasma cell. Here it is. Okay, first exposure. Okay, so the allergen, they produce the MSC molecules on the surface and toll-like receptor binds to the B cells, and the Th2 T cell helper cells, okay, helps to differentiate into the memory cells and plasma cell. And the plasma cells secrete the IgE antibody. The secreted IgE antibody binds to the IgE specific Fc receptor, the Fc receptor present on the mast cell surface, and also blood basophils. Now here the second exposure to the allergen leads to cross-linking of the bound IgE, triggers the release of pharmacologically active mediators. Here it is. This is antibodies and Fc receptor. Okay, they sensitize mast cell. They produce the allergens. Okay, second exposure of allergen, they leads to degranulation. The mediator caused, they produce the mediator, they pharmacologically active compounds like vasoactive amines from mast cells and basophils. Okay, so histamine, they are the vasoactive amines. The mediator cause the smooth muscle contraction, okay, increase vascular permeability and vasodilation. Okay, so how the degranulation occurs. Okay, exposure of allergen and IgE binds to the Fc receptor present on the mast cell and basophils. Okay, now IgE actually binds to different receptors. First, the high affinity Fc epsilon R1, which is responsible for the most of the symptoms we associate with the allergy. Second, that is the low affinity Fc epsilon R2, which is regulate the production of IgE by B cell as well as transport across the cells. Right? Also, interestingly, the serum half-life IgE is quite short, only 2 to 3 days. However, when bound to its receptor on innate immune cells, IgE is stable for weeks. Okay, so it is a different the half-life of the IgE. Now, the high affinity of IgE receptor Fc epsilon R1. Right. So, mast cell and basophils constitutively express high level of the high affinity IgE receptor. However, low level of IgE receptor, that is the Fc epsilon R1, expressed on the eosinophils, Langerhans cells, monocytes and platelets. So here it is the structure of high affinity IgE receptor Fc epsilon R1. So most cells express the tetrameric form 1, 2, 3, 4. Okay, tetrameric form Fc epsilon R1, which includes an alpha, beta, and 2 identical gamma chain. This is linked by the disulfide bond. Here it is. Now another cell, that is the monocytes and platelets, express an alternative form lacking the beta chain. Okay. The alpha chain, here it is the alpha chain of the Fc epsilon R1, member of the immunoglobulin superfamily, direct binds to the IgE heavy chain. Okay, here it is the IgE-like domain. They binds to the heavy chain of the IgA. Where is the beta and gamma? Okay, chain are responsible for the signal transduction. So they contain the ITAM, immunoreceptor tyrosine based activation motif, that are phosphorylated in response to IgE cross-linking. Okay, so this is a structure of high affinity IgE receptor, that is a tetrameric. Now another, that is the low affinity IgE receptor. So low affinity IgE receptor, or we can say the CD23, is much lower affinity for IgE. Okay, and CD23 structure is distinct from Fc epsilon R1. It is lectin and type 2 membrane protein. So what is the type 2 membrane protein? The transmembrane helix in the type 2 membrane protein is typically hydrophobic. It is a hydrophobic part, okay, and span the lipid bilayer. Okay, unlike type 1 membrane proteins, which have their end terminus outside the cell. Okay, in type 1, end terminus outside of the cell and the C terminus inside. But type 2 membrane proteins have the opposite orientation. Okay, so this is the end terminal cell inside the cell and outside the C terminal. Okay, so end terminus cytoplasmic and C terminus extracellular lumen. Okay, and existence of two isoforms that differ only slightly in the end terminal cytoplasmic domain. End terminal. There is a two isoform. Okay, just differ from the end terminal of the cytoplasmic domain. That is a CD23A and CD23B. Okay. So CD23 is present on activated B cells, while the CD23B is produced on different cell type in response to the signaling molecules interleukin 4. So both isoforms exist in membrane bound and soluble form. The soluble form are created when the surface molecule is broken down through the proteolysis. Interestingly, CD23 binds not only to IgE, okay, but also binds to the complementary receptor CD21. Now the outcomes of the CD23 ligation, okay, depends on the which ligands it binds to IgE or CD23 and whether it does so is soluble form or membrane bound molecules. So this is the these molecules. There is two isoforms, CD23A and CD23B. It is differ only the end terminal parts. Okay. So CD23 ligation depends on the ligands binds to the either IgE or CD23, that is a part of the IgE domain. Suppose when soluble CD23, binds to CD21 and on the surface of the IgE, okay, synthesized B cells. So the IgE synthesis is increased. However, when membrane bound CD23 binds to soluble IgE, further IgE synthesis is suppressed. Okay, so here it is the difference how the CD23 binds to the different ligands. For example, they binds to the CD21 of IgE. Okay, they increase the IgE synthesized, but they bound the soluble IgE. Okay, and then suppress the IgE synthesized. Okay, so IgE antibody binds to the Fc epsilon R2, which are present on the B cell surface, which is only present on the B cell surface, and which binds to the CD23 soluble form. Okay, IgE. So the IgE synthesis is suppressed. If they bounce to the CD23 with the CD21 of IgE, so IgE synthesis increased. Okay, so this is the depend of CD23 ligation depends on the which ligands it binds to. Okay, so people with the allergies tend to have higher amount of both the attached and soluble form of CD23. Okay, so this is all about the type 1 hypersensitivity. Okay, the receptor, the mainly main role, that is IgE, okay, which is responsible for the hypersensitive one or producing allergies. Okay, so in short, that IgE binds to the Fc epsilon R1 receptor, which are present in mast cell and basophils, and create the cascade of events and deregulation of mast cells, and produce the pharmacological compound, vasoactive compounds, which attack or role of the particular organ, like construction of muscles. Okay, so now the questions. These are the multiple choice questions. Okay, first, what is the main effector cell involved in type 1 hypersensitivity reaction due to the IgE binding? So effector is the main effector cells which produce the primary mediators, secondary mediators, or we can say the degranulation. Okay, so it is the simple answer is mast cells. Now, which immunoglobulin is predominantly involved in hypersensitivity type 1 reaction? Okay, so type 1 always involve the IgE. What is the primary cellular mediator in immediate hypersensitivity reaction? Okay, primary cellular mediators. Okay, also answer is the mast cell. In allergic rhinitis, hay fever, exposure to allergen leads to the release of mediators causing. Okay, so that is the very simplest answer, that watery eyes and sneezing. Which of the following common routes of allergy export in type 1 hypersensitivity? It is inhalation. Now, which of the following is the common term for the IgE binding receptor mast cells and basophils involved in type 1 hypersensitivity? Which receptors are present on the mast cell and basophils? Okay, so there is a Fc epsilon R1. Okay, that is the CD20 not, CD4 not, or Fc gamma not. Okay. Now, in hypersensitivity type 1 reaction, the IgE binding receptor primarily found on. Okay, so IgE binding receptor. Okay, that is the mast cell and basophils. Okay, so this is all about today's video, and I hope it was helpful. Thank you for watching.